So-called molecular scissors. These are restriction enzymes, and there are vectors. The plasmid DNA acts as vectors, okay, to transfer the piece of DNA. Okay, there is cloning. Okay, like the ability to multiply copies of antibiotic resistant gene in E. coli was called cloning of antibiotic resistant gene in E. coli. So this is cloning. Now, tools of recombinant DNA technology. So tools of recombinant DNA technology. First one is restriction enzymes. You see, restriction endonuclease. There are recognition sequences. First restriction endonuclease hinder 2. Okay, nucleases are restriction enzymes belonging to a larger class of enzymes. Nucleases. Now, these are two kinds of nucleases: exonucleases, endonucleases. This is why for restriction enzyme we call restriction endonuclease. Endonuclease. Please. There is palindromic nucleotide sequence in the DNA. Each restriction endonuclease recognizes a specific palindromic nucleotide sequences in the DNA. Now, steps in formation of recombinant DNA by action of restriction endonuclease enzyme EcoRI. So, action of restriction enzyme, you see, the enzymes cuts both DNA strands at the same sites. Let's take the vector DNA. Okay. Now on the other side, EcoRI cuts the DNA between bases G and A only when the sequence is GAATTC, which is palindromic sequence, is present in the DNA. You see, foreign DNA. Now these are cutted, and then both produces sticky ends, foreign DNA and vector DNA. Now DNA fragments join at sticky ends. You see, TTAA, AATT. Now recombinant DNA is formed. Let's see the steps in formation of recombinant DNA by action of restriction endonuclease enzyme EcoRI. You see now diagrammatic representation of recombinant DNA technology. Now foreign DNA, vector DNA. You see the plasmid. Now foreign DNA, same restriction enzyme cutting both foreign DNA and vector DNA at specific point, and then ligases join foreign DNA to plasmid, then transformation into the E. coli cell, then cells divide, then multiple copies are formed. There is process, that is separation and isolation of DNA fragments by gel electrophoresis. A typical agarose gel electrophoresis showing migration of undigested lane 1 and digested set of DNA fragments lane 2 to 4. You see, largest near the wall wells, the smallest farthest. They move large distance. Anode is positive, DNA is negative. Okay. Exposure to UV radiation and stain with ethidium bromide, then exposed to UV radiation. Bright orange color bands